Thank you, Adora. Good morning, everyone. So I'm Anne-Virginie Eggimann. Most people call me AV, so feel free to do so. Thank you, Adora and Rick, for inviting me. I'm very excited to be here to share with you the story of our lentiglobin BB305 drug product for the treatment of beta thalassemia. So this morning we talked a fair amount about how you go from research to starting your first clinical study to IND. I would like you to think about zooming out a little bit, and what I'm about to walk you through is a little bit of a longer journey, starting from the research, but going all the way to marketing approval. And being head of regulatory, I will obviously focus on regulatory aspects, and there's a lot of acronyms in regulatory, so I apologize in advance because I'm sure I didn't catch all of them on my slides. So this has been a long journey, and for me it actually started in the late 90s, early 2000 in Paris when I got my first job in regulatory. That's when Alain Fischer, Marina Cavazena were conducting the XGID clinical trials, and that really started my passion for gene therapy in starting my career, and I could see firsthand the potential of gene therapy as well as the safety aspects, which we all need to be mindful of. And fast forward 10 years later, I met our CEO, Nick Leshley, actually in DC at a cell and gene therapy conference. A lot less people at the time, it wasn't as fancy. And then 10 years later, here we are. And as Adora mentioned, we did receive some good news in Europe, where we received what is called CHMP positive opinion. So that is an opinion from the European Medicine Agency, which is a scientific opinion on a marketing authorization application. The next step after that is for the European Commission to grant a decision and to grant marketing authorization, which we're expecting sometime this quarter. So the EMA recommended a conditional approval for lentiglobin BB305, which is now called Zynteglo. I still have to get used to that. For the treatment of patients who are 12 years and above and who have a non-beta 0 beta 0 genotype and who do not have an HLA match sibling match-related donor. Sorry, I'm having trouble with this. So some details on the opinion. First, and we'll talk a lot about some of how we were able to accelerate the development of this product. And in part, they note in their press release, the agency, that we benefited from PRIME, so I'll tell you a little bit more about that. But this is a conditional approval, and basically the key point here is that this approval was early in the development. So if you think of traditional development, you have phase 1, 2, and then phase 3, and usually you wait for the results of phase 3, and then you file, and then you get approval. So we were able, through mechanisms that I'll share with you, to obtain conditional approval based on early phase 3 data, so which is highlighted here. And the last thing that's highlighted on this slide is the agency is then clear that the next step after that to grant patient access is to go through the price and reimbursement process in each country. And I'll speak a little bit about that because part of the interactions we had with regulators involved what we call HTA bodies, Health Technology Assessment bodies, who are in charge of pricing and reimbursement. So how did we get there? So before I go into the specifics of our case study, I wanted to share a little bit sort of our guiding principles in regulatory at Bluebird and how we operate, and I'm hoping that will be useful. So first, the definition of regulatory science, and this comes from Hans-Georg Heischler at EMA. Regulatory science should enable the co-evolution of science and regulation. And what I really like about this definition is the word enable. And at Bluebird, we see regulators as partners. We don't see them as a hurdle, but we see them as partners throughout the development. And I think because we're lucky to work in a very innovative space, they are very engaged because they want to learn along our journey as well. So regulatory science can evolve at the same pace as science, which is tricky. I think they're a little behind. But this concept, generally speaking, is very motivating, at least on my side, to be engaged and to be here and to share, and I'll speak to that. So regulatory science, you can also see it basically as a bridge between the company and the regulators. And your job is to translate the great science that you guys do and make it a clear story for the regulators and vice versa. So some of our guiding principles is to be transparent and proactive in our communication. Where I think if you're going to convince the agency of anything, it has to be science-based, data-driven, and patient-focused. I think that's very, very important. Don't be afraid of lack of precedent. I think that's less and less relevant, but when we started about 10 years ago, there was a very little precedent. No gene therapy products were approved. So that was very important for us to really be creative in thinking about our path forward. And part of this thinking is to really be engaged externally with the agencies as well as other stakeholders, bring this learning into our own development, as well as share the learnings from our development externally. So it's a sort of a virtuous circle, which I think works very well and actually helps after you have your IND and you want to look at this marketing approval. Being engaged in the regulatory science fields, I think, can help a lot. And how are we thinking in general? Well, there's always 3 buckets when you're in regulatory. There's CMC, non-clinical, and clinical. So whenever I speak to anyone, I always tell them, which bucket are you in? And the important thing I wanted to highlight is I think in our field, CMC and clinical are extremely related, much more so than if you work on a small molecule where it's completely different. And I think if you want to be successful, you have to be able to understand all that, which is hard. Often people focus either on CMC or on clinical or on non-clinical. But I think it's important to really think holistically, particularly for autologous products. I think also you have to think globally. We were very fortunate to be able to manage all our regulatory interaction in the US and in the EU. And I don't know if I'm going to be able to tell this joke well, but if you're familiar with Asterix, you know, he fell in the little cauldron where they made the magic potion very early on. I kind of feel that way because my mom is French and my dad is American, so I always think EU-US. And so it's very easy for me to think this way. But I think some companies focus very much on the US and sort of forget about the EU. And I think there's a lot to learn in both interactions. I highly recommend considering both very early on. And another key concept is, and this is a very busy slide, but the key concept is if you go from very early development all the way to marketing authorization and beyond to post-approval, this is a long journey. And I think the best way to start the conversation with regulators is to actually share your vision for this entire life cycle. That's not easy. Regulators often like to start talking about phase 1, and then once you have those results, they want to talk about phase 2, and then they want to talk about phase 3. It's very important to start laying out your plan so you can start getting feedback on your vision. And part of the fundamental, I would say, principle that really impacted our development was the concept of adaptive licensing, which was then referred to as adaptive pathways. There are 2 key publications that explain these key concepts by primary author is Eichler, and Gigi Hirsch from MIT New Dix is the senior author on these publications. And Dr. Eichler was in secondment at MIT, and he tried to think about how can we bring medicinal products that are promising to patients earlier. And he developed this concept of, okay, what if we agreed with all the important stakeholders on the entire evidence generation plan throughout the life cycle of the product? So very early on, you talk with regulators, with patients, with HTA bodies, with payers, and what if we agreed on that development plan? Wouldn't that be then possible to get an earlier approval? Because you would know with a higher level of uncertainty, but you would then know that the evidence would continue to come. And some of the concepts there which are familiar in oncology would be maybe to start with a smaller indication and you expand over time. Also the idea of if you're on the market, instead of conducting a trial, you're going to use real-world evidence. So that was also a fundamental concept of trying to have an earlier approval and then build registries that will then be able to collect additional information post-approval. And I talked about early involvement of health technology assessment, and I'll speak a little bit more about that. And this is just a quick picture of all the stakeholders we interacted with. And I want to note, I spoke about MIT and UDIGS. We did a few scenario design sessions with them. So this is a session where you come in with your case study and you have a fair amount of people involved from industry, from academia, from regulatory bodies, from payers, both in the US and EU. And you walk through the different scenarios that you could use throughout the development of your program. And so we did this a couple times, and that was very, very helpful. We are also very engaged with the Alliance for Regenerative Medicine, and we had advocated for the role of the RAC to evolve, and we were very happy to see that. We also worked very closely with the FDA to develop RMAT, and to emphasize the importance of early interactions, which then turned into the Interact meetings. So these are just some examples of what I've been very fortunate to be able to be part of at Bluebird, is also engage and shape our environment. So the case study, I have one slide that has a lot of busy information, but maybe I wanted to tell you a quick few things about beta thalassemia. So it is an inherited blood disease that requires lifelong frequent blood transfusions and iron reduction therapy. We conducted 2 phase 1/2 studies and 2 phase 3 studies. In patients with TDT, so these patients require transfusion to be alive, and they have impaired beta globin production, which results in excess of alpha globin chains. They have severe anemia and require transfusions, as I mentioned, which then causes a lot of iron overload and serious comorbidities. These are the studies that we conducted, and I mentioned earlier that we also are planning to collect real-world evidence, so we are partnering with existing registries, patient registries, to collect information post-approval. We also have a long-term follow-up study for 15 years follow-up for all the patients included in the trials. So this slide is about our journey, and there's a lot of information here. But I first wanted to focus on the beginning of the journey. So in 1994, Philippe Lebouche created the first modified human beta globin with anti-cycling properties. And then in 2001, created the first LVV with this modified gene. And there are a fair amount of publications that came out of that. I will go through them very quickly. I think hopefully some of you saw the presentation by Dave Davidson earlier this week, and he went through some of these details. So here was the first publication to show the impact of the gene therapy in mice that could basically correct the beta thalassemia phenotype. In 2010, the first publication was by Marina Cavazana, and this was with the first version of the LVV, which was called lentiglobin HPV 569. And you can see on the right that from the transduction of the CD34 cells that then engrafted in the patients, we were able to produce 3 grams per deciliter of what we call beta 8087Q, the transgene hemoglobin. And so when we started the development, we thought, oh, maybe we could get to 5 grams. And today, we have actually improved the vector, and Dr. Veresh was involved in doing that, and we created lentiglobin BB305 drug product. And now with our patients, we're able to produce up to 10 grams per deciliter of transgene. So that gives you an idea of the scientific journey that we went through. And that's the design of the vector. And the good thing about having this anti-cycling property with the threonine to glutamine substitution at the position 87 is that we can measure the protein in the blood. And I think if you think about accelerating your development, I think this has been very helpful because regulators can see the impact of the product and the patient. It's very easy. Sometimes it's not always easy depending on the disease you're treating. So going back to our journey, I walked you through the early scientific aspects. I joined Bluebird in 2011, so our first job was really having pre-IND meetings, pre-CTAs. So CTAs are clinical trial applications in Europe. So we had interesting meetings with Philippe, talking to FDA and walking him through his great science. It was very exciting. We then filed an IND, I think in December 2012, and then we treated our first patient, actually in France, in 2013. So shortly thereafter is when the EMA came up with their adaptive pathway pilot. And we were the first company, or one of the first company, to apply. And I walked you through the theoretical of that pilot, and I know that may be complicated. But the key thing that we were able to do with this pilot is have a lot of interactions with the EMA in what they called safe harbor discussions. So typically when you go to the regulators, you go with a plan, and then you ask questions, and then they agree or disagree, and then you move forward. And if they disagree, sometimes you can get a little bit more color, but you still have to come back with a different plan. Within this pilot, we were able to have discussions around scenario planning. So we were able to say, okay, well, we could use transfusion independence as an endpoint, or we could use transfusion reduction, and we could enroll this many patients or this many patients, and here would be the different assumptions. And so it was very helpful very early on to have those discussions, and the part of the pilot was to have in the room patients, regulators, and again, HTA buddies. And if you think about where Bluebird was, I don't know, we were 20 when I joined, perhaps we were 150 or something like this, I don't know, we're about 800 now. But these early discussions about what it would be like to be on the market with the HTA buddies really, really transformed the company. We started thinking about where we are today in 2014. So that was, I think, very, very helpful. And we received a lot of feedback during these interactions. And as some of you know, possibly, the agency then took all the learnings from this pilot. So we were one of the company. I think there were about 10 companies that participated. And then they translated this process into what they call PRIME, which is Promising Innovative Therapies. And in the US, the FDA also created Breakthrough Therapy Designation, which is slightly different, and we'll talk about that. So we were able to benefit from these designations. In parallel, so I talked about modifying the vector. During our phase 1/2, we also realized that we needed to increase the amount of protein produced for our patients to become transfusion independent. So we refined the drug product manufacturing process. And so we had a lot of discussions about comparability, which in our field is quite complex. And within discussions that we have with DORA, within ARM and other association, we talk a lot about similarity, exclusivity, what is the same, what is not the same. And I think it is going to be a long time before we can completely figure that out, but it is very interesting. And then we submitted the MAA in 2018. We were able to have accelerated assessment, and then after the accelerated assessment procedure was pretty intense. We had a 1-month clock stop, which means we got the questions and we had to respond in 1 month. So that was pretty intense. And overall, benefiting from PRIME and Breakthrough and all these specific designations and interactions with the agency has been really helpful to accelerate the clinical development of the product. However, the challenges are more on the CMC front. You know, we talked about validation, we talked about all the things we need to do to support marketing approval, and shortening time frame for these activities is very, very challenging. You typically, at least we do, work with contract manufacturing organizations, CMOs, and there's a lot to do, and it's really hard to squeeze the time. So I think that's an area where in our field we can all try to be creative and come up with new ideas, including on the regulatory side of what could be some of the mechanisms to accelerate the CMC development. And just a quick note, so we submitted the MAA in 2018, and we anticipate submitting the BLA in 2019. So you can see that it is about a year difference. So I was asked to kind of share my thoughts about the similarities and differences between the US system and the EU system. I think overall it's very similar. We've had great interactions with the FDA under breakthrough, as well as with the EMA. I think the main difference, people ask me all the time, why are you submitting in Europe, and why are you going to be approved in Europe first? I really think it was because of this pilot and these early interactions that we had, where we were able to really have a lot of dialogue. I think the agency was very, very familiar with our product and very comfortable with the path that we had laid out. And I am positive that things will go well with the FDA as well, just a little bit later. So concluding remarks on the regulatory process, I think you've heard many times, be engaged. I think it is good to shape your development and your environment. Chance favors the prepared mind. I think when the EMA came up with their pilot program, we were ready because we knew it was coming. We knew that for years, EU regulators were trying to think about how to accelerate access for medicinal products that were promising. So being engaged, knowing what's happening. I also recommend commenting on draft guidelines. We talked a lot about guidelines this morning. On the FDA website and the EMA website, they have a fair amount of guidelines that are available for commenting. For example, right now the EMA has a guideline on quality, non-clinical, and clinical requirements for investigational advanced therapy, so clinical trials. For example, I think it would be great if we all provided comments. I think the requirements would then be even better and more adapted. Be collaborative and have early interaction with all the stakeholders. We learned a lot from interactions with patients very early on in terms of trial design and endpoints. I think being patient-focused in your decision-making is also very helpful. It helps you go in the right path. And then take advantage of expedited pathways and think early about your entire life cycle. So I only have a few quick more slides about what's next for us. So I mentioned that we are planning to file in the US. I also mentioned that our first approval was for patients with non-beta 0, beta 0 genotype. So we are planning to expand to patients who have a beta 0 genotype, as well as pediatric patients less than 12 years of age. So we have a little bit more work to do, which is this concept of iterative extension of indication. Start early, but then you can expand. And then in the EU, we're gearing for launch, and we want to get it right. So we are working on manufacturing, making sure we have our registries set up, having our commercial team work well with the physicians and the patients, and then our supply chain. So part of what's next is to interact with what we call the qualified treatment centers. So these therapies are going to be delivered in specific centers. I don't think this is specific to us, but there's a lot of work there. And also we are starting discussions with health technology assessment bodies in Europe. As you can imagine, getting price and reimbursement for a one-time therapy that is potentially curative is new for them. And our CEO has shared earlier this year that we are planning to propose a value-based, outcome-based contract where payment would be over time, over 5 years, and in this case it would be based on the need for transfusions. So I wanted to thank a lot of people because this journey obviously has been a long journey, but I definitely want to thank Laurice. She's a patient who has been involved with us in many of the Design Labs sessions. Not many, but the few Design Lab sessions that we had. She's a patient advocate in California, and She was very helpful in making us understand the disease and what it means for her to have thalassemia. So I'm very thankful to her, very thankful to all the patients and families who contributed to our clinical trials, as well as many, many bluebirds. So Dave Davidson, who is my boss, our Chief Medical Officer, Nick Leshley, our CEO, who hired me and took a chance, Philippe Lebouche, Marina, and Jean-Antoine, who created the vector and treated the first patient with beta thalassemia. I also want to thank people on my team, Eleanor, Jesse, Leslie, Sarah, Andreas, and Claire, and many, many more bluebirds. Thank you so much.
Can you hear me? Okay. Thank you, AV. That was a really very informative presentation, and congratulations on your development. We'll take one or two questions from the audience.
Nice presentation, AV.
Thank you.
Much success. Thank you. I have two quick questions for you. One is, what were the unanticipated bumps in the road that you had to overcome that came up unexpectedly during this process? Mm-hmm. And number two, could you care to describe your meetings with the PDCO for the Pediatric Investigational Plan and follow-up?
Yes. Okay. So bump in the road, I think one comes to mind that was very early on. Actually, my first week at Bluebird, we had just transferred the manufacturing process for our lentiviral vector, and we had just found out that titer was 0. So that was the first, my first week, and that was an issue. So, we put a team together and worked very hard to figure out what happened. And I think what we learned from that first bump in the road is that, yes, you're going to have many of those. And as we talked today, if you're resilient and you really try to go through it, you're going to learn a lot. And you're going to be way better and way ahead. So that was one example. I think at some point we had some issues with mice that developed tumors. And so we had to figure out, was it related to our vector or was it just related to the mouse model? And we were able to show that it was related to the mouse model. But that was also a good 3 to 6 months of intense work. And that was very early on. I think later on, the biggest challenges that we've had has been more on the manufacturing side and trying, like I said, to really match what we're doing on the clinical with CMC. And we've had a lot of changes. We've in terms of processes, as well as in terms of manufacturers. So, we have a manufacturer in the EU and a manufacturer in the US. The manufacturer in the US that did the clinical trials had a different facility for commercial, so we had to transfer. So it's a lot of changes, but I would say now we're really used to it, and we're getting better. So I think in terms of unexpected bumps and issues, those were the big challenges. I think, also you never know when you start your clinical trial what your data is going to look like. And so we've had to optimize our product to be able to produce sufficient protein. So I think those are the main thing. And I'm sure we're going to learn a lot through the HTA process, and I'm sure it's not going to go 100% smoothly. And your question about the pediatric development plan. So I know that they can be challenging sometimes for sponsors. For us, it went relatively well. And we were able to, for example, initiate scientific advice and then pause, go through the pediatric procedure, and then finish scientific advice. And that went relatively smoothly. The scientific advice working party and the PDCO were aligned. And as you could see, our first approval is for 12 years and older, so both FDA and EMA recommended staggering the development. So we started with adults, then adolescents, and then pediatrics. So I think that the PDCO was supportive. They did ask us to consider patients very, very young in the development. And I think they had a bone marrow transplanter on the committee who suggested we include patients who are 6 kilograms at a minimum. So that was kind of a surprise, for example. But, overall it went well. I guess. I'm happy to answer more questions on that.
Thank you, Avi, and I'm hoping that you're going to stick around after because I'm sure there are a ton of questions. We are running behind, so if I could ask those that have questions to maybe catch Avi after the session. Thank you very much for a really phenomenal presentation.